Welcome to Understand All. I'm your host, Aslakir Gerleaf. Today, we're embarking on a fascinating journey into the world of CRISPR, a revolutionary gene editing technology that's redefining the boundaries of science. So, buckle up and get ready to have your mind blown. Picture this: a world where we can edit our DNA like a document, correcting typos and rewriting the very code of life. That's the promise of CRISPR, and it's not just science fiction anymore. In fact, according to a 2021 report by Grandview Research, the global CRISPR technology market is projected to reach a whopping $6.7 billion by 2027, up from $1.2 billion in 2020. Let that sink in for a moment. But what exactly is CRISPR and how does it work? Well, CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which is a mouthful, I know. But bear with me. It's essentially a gene editing tool that allows scientists to make precise changes to DNA with incredible accuracy. In fact, a 2014 study published in Nature Biotechnology found that CRISPR-Cas9 can target and modify specific DNA sequences with an astonishing 99% accuracy. That's like hitting a bullseye from miles away. Now, you might be wondering, how did this game-changing technology come about? It all started with a curious observation in bacteria. Researchers noticed that bacteria had this weird way of defending themselves against viruses by cutting up the viral DNA and storing bits of it in their own genome. It's like they were keeping a genetic mugshot of their attackers for future reference. Fast forward to 2012, and the DNA dynamos, Jennifer Doudna and Emmanuel Charpentier, cracked the code. They figured out how to harness this bacterial defense mechanism and turn it into a precise gene editing tool. And just like that, the CRISPR revolution was born. But how does CRISPR actually work? Picture a pair of molecular scissors guided by a GPS. The GPS in this case is a piece of RNA that matches the DNA sequence you want to edit. When the RNA finds its target, the scissors, an enzyme called Cas9, snip the DNA at that exact spot. Then, the cell's natural repair machinery takes over and can either disable the gene or insert a new piece of DNA. It's like fixing a typo in the book of life. Now you might be thinking, that's cool and all, but what can we actually do with this technology? Oh, my friend, the possibilities are endless. Let's start with medicine. CRISPR has the potential to revolutionize the way we treat genetic disorders. Take sickle cell anemia, for example. It's a debilitating disease caused by a single genetic mutation. But with CRISPR, we could potentially correct that mutation and cure the disease altogether. In fact, CRISPR has already shown promise in clinical trials. According to a 2021 study published in the New England Journal of Medicine, CRISPR-based gene editing was successfully used to treat sickle cell anemia in patients, with some even achieving complete remission. That's not just a breakthrough. It's a life changer for millions of people worldwide. But the medical applications don't stop there. CRISPR is also being explored as a powerful tool in the fight against cancer. By targeting and destroying cancer-causing genes, CRISPR could potentially offer a new avenue for cancer therapy. It's like having a cancer-fighting superhero on our side. And let's not forget about infectious diseases. CRISPR has shown potential in targeting and eliminating viral infections like HIV and hepatitis B. In a 2017 study published in Nature Communications, researchers used CRISPR to successfully eliminate HIV from the genomes of living animals. While there's still a long way to go, this is a promising step towards a world without devastating viral diseases. But the CRISPR revolution extends far beyond medicine. In agriculture, CRISPR is being used to develop crops that are more resilient, nutritious, and environmentally friendly. Imagine crops that can withstand droughts, resist pests, and even have enhanced nutritional value. It's like giving plants superpowers. In fact, a 2018 study published in Nature Communications found that CRISPR-edited crops could have yields up to 50% higher than their conventional counterparts. That's not just a boost for farmers, it's a potential solution to global food security challenges. And let's talk about the environmental applications of CRISPR. From conservation efforts to bioremediation, CRISPR is proving to be a versatile tool in the fight against environmental degradation. Researchers are exploring ways to use CRISPR to protect endangered species, control invasive species, and even clean up pollution. It's like having a gene genie for the environment. But with great power comes great responsibility, and CRISPR is no exception. The technology raises important ethical questions that we, as a society, must grapple with. Designer babies, human enhancement, unintended consequences. These are all valid concerns that require careful consideration and robust public discourse. And let's not forget about the accessibility and equity issues surrounding CRISPR. Will this technology be available to everyone, or will it exacerbate existing health disparities? These are questions we must address as we navigate the uncharted waters of gene editing. But despite the challenges, the future of CRISPR is undeniably exciting. Researchers are pushing the boundaries of what's possible, exploring new applications, and refining the technology every day. Who knows what breakthroughs await us in the coming years? Maybe we'll see CRISPR-based therapies for Alzheimer's, Parkinson's, and other neurodegenerative diseases. Maybe we'll have crops that can capture carbon from the atmosphere and help combat climate change. Maybe we'll even uncover new secrets of human biology and evolution along the way. The possibilities are truly endless, and that's what makes the CRISPR revolution so thrilling. We're standing at the precipice of a new era in science, one where we have the power to rewrite the code of life itself. But as we embark on this journey, let's remember to proceed with caution, compassion, and a deep sense of responsibility. Let's engage in open and honest conversations about the ethical implications of gene editing. Let's work towards a future where the benefits of CRISPR are accessible to all, not just a privileged few. And most importantly, let's never lose sight of the awe and wonder that drives us to explore the mysteries of life in the first place. Because at the end of the day, CRISPR isn't just a tool. It's a testament to the boundless curiosity and ingenuity of the human spirit. So, as we venture forth into this brave new world of gene editing, let's do so with eyes wide open, minds engaged, and hearts full of hope for the future. The CRISPR revolution is just beginning, and I, for one, can't wait to see where it takes us. And that, my friends, is the story of CRISPR, the little bacterial defense mechanism that could and did change the world. I hope this journey has left you as awestruck and inspired as I am. But don't just take my word for it. Go out there and learn more about CRISPR. Engage in discussions, ask questions, and form your own opinions. Because the more we understand about this incredible technology, the better equipped we'll be to harness its potential for good. And who knows, maybe one day you'll be the one leading the charge in the next great scientific revolution. Until then, stay curious, stay engaged, and never stop exploring the wonders of the world around us. Thank you for joining me on this adventure into the world of CRISPR. I'm your host, Aslakir Gareleaf, and this has been Understand All. Until next time, keep asking questions and seeking answers. The future is ours to shape, one gene at a time.